Hi everyone, I'm going to talk about how we can use phylogenetic analysis to resolve evolutionary relationships. So this is an outline of what I will discuss. When we talk about phylogeny, we need to know how to read one of its most important data representation, which is a phylogenetic tree. A tree represents evolutionary relationships among biological entities, such as species, genes, or proteins. We call them operational taxonomic units, or OTUs. Each leaf is an OTU. A node represents common ancestors of the OTUs above it, and a root is the common ancestor of all OTUs. So why we need a tree? One of the reasons is because we want to find a natural way to arrange organisms and resolve their relationships. We classify organisms in many ways. For example, plants are edible or inedible, but these are artificial classifications. Modern taxonomists prefer natural classification that reflect true evolutionary pathways, and phylogenetic trees help achieve this. A good taxon should be monophyletic, which includes the most recent common ancestor and all its descendants. For example, human plus great apes and our most recent common ancestor form a monophyletic group. This makes one very clean cut on the tree and takes away all branches above the cut. Less desirable groupings include paraphily and polyphily. A paraphyletic group includes the most recent common ancestors, but excludes some descendants. A classical example is the traditional reptiles, which excludes birds, although birds are the closest living relatives of crocodiles. This makes reptilia paraphyletic. Polyphyletic groups have mixed phylogenetic origins and do not include their most recent common ancestors. Most biology students must have learned the Cronquist taxonomic system for plants. Cronquist used to put water lily, lotus, and hornworts inside the same family called Nymphaceae. So where are these plants on a phylogenetic tree? So this is a part of the APG phylogenetic system, representing some of our latest belief of flowering plants relationships. Water lily is here, lotus is here, and hornwort is here. So they are from mixed origins. So we can say that Cronquist Nymphaceae is polyphyletic. You may have heard about a concept called waste-bucket taxon, where organisms of unresolved relationships are all thrown inside. This is also an example of polyphily. So how can we perform phylogenetic analysis to build a tree? First, we select the sequence of interest. Selection depends on the subject we want to study. If we are investigating some recent evolutionary changes, we can choose a sequence that has fast evolutionary rate. Else, we should choose more conservative sequences such as rRNA. Then, we identify homologs, which are sequences in different organisms with the same origin. Then, we align the sequences, and finally, we calculate the phylogeny. There are three schemas we will discuss: parsimony, distance, and maximum likelihood. So let's first take a look at maximum parsimony. The method follows an idea called Occam's razor, which states that when all other factors are equal, the simplest explanation should be chosen. In our case, we prefer the tree requiring fewest evolutionary changes because we assume such changes are rare. Let's take a look at these two trees built from the same four OTUs. Topology of the first tree only requires one evolutionary change, while the second tree requires at least two. Therefore, we should choose the first tree because it is the most simple tree. Now that we know how to compare trees, how can we find the best tree? The naive method is to first enumerate all possible trees, then calculate the number of changes required by each tree. Finally, we select the tree with least number of changes. So you may notice that number of trees grows exponentially with the number of OTUs. This makes the naive method extremely inefficient. Tree searching algorithms such as nearest neighbor interchange address this issue by iteratively modifying topologies to find more parsimonious tree. We do not need to enumerate all possibilities in this case. So do we have a faster method than searching? And the answer is yes. Distant methods are faster by progressively clustering OTUs. It does not need to enumerate possibilities. So examples of the method include UPGMA and neighbor joining. We start with a distant matrix, repeatedly join the closest pair, update distances, and build the tree. So here is a UPGMA example with 6 OTUs. We first find the closest pair, which is A and B. We join them, and we update the distance. So now D and E is a closest pair. We join D and E, and we continue these procedures until we complete. So the process is quick without enumerating trees, but UPGMA assumes constant evolutionary rates. If we want to relax this assumption, we can use neighbor joining methods. Finally, we can take a look at the maximum likelihood approach. This requires us to find a tree with the largest likelihood. We toggle parameters of the tree to maximize the probability. This resembles gradient descent, optimizing towards the highest likelihood. So how can we calculate the probability given some parameters? I will give you a toy example. So in this example, with 4 sequences, we calculate the probability of each column Lj by summing over all possible ancestral states as shown in this equation. To calculate the probability of each ancestral state, such as the state shown in this figure, where both nodes are A, we calculate the transition probability from one arbitrary node, in this case C, to the rest of the nodes. Finally, slight likelihood Lj are multiplied together to get the overall likelihood of the alignment given the tree. If you want to avoid multiplication leading to very small numbers that are very hard to represent on a computer, you can log transform the likelihood. Finally, we can see that previously all trees do not have a root. We can root the trees by including an alt group. Alt groups are OTUs known to have diverged early from the groups of interest. So after all this theory, we can take a look at an example. Suppose we know that true crabs have marine ancestors, but some lineages, like this crab in the image, which is endemic to Hong Kong, live in fresh water. Suppose we want to know how many times did crabs evolve freshwater habitation. So to answer this question, researchers at CUHK reconstructed crab phylogeny. You can recall that phylogenetic analysis can be divided into 4 main procedures. So the researchers first sampled multiple groups from the crabs. They identified 8 genes to use as homologs for analysis. They aligned the sequences using muscle, and they used some shrimps as outgroups. Finally, they calculated phylogeny using maximum likelihood approach. So this is the result. This part of the phylogeny tree shows primary freshwater adaptation occurred at least twice and possibly 3 times. You can see from the highlighted figure that there is one paraphyletic group, mainly in the Old World, and one monophyletic group in the New World. So in summary, phylogenetic analysis infers relationships through common ancestry. It reveals evolutionary histories. It can track pathogen spread such as COVID-19 during outbreaks. And it can also help discover new sources of bioactive compounds such as taxol by identifying closely related species that are likely to share useful traits. And that's all for my sharing. Thank you for listening.